this DNA replication and transcription and translation and all that sort of thing, and get that down pat. It's the basics of who you are. It's the basics of your DNA and how it works. I want to show you a little video before we get started. Okay, this might actually pique your interest a little bit, and at least it will sort of help me, because it's with motion and action, it's a little easier to see this process happening. So, let's see what happens here. Yeah. That's the problem. I want to start this over.
Sure. The process starts when DNA is wrapped around special protein molecules called histones. The combined loop of DNA and protein is called a nucleosome.
We're going to talk about that.
Next, the nucleosomes are packaged into a thread. The end result is a fiber known as chromatin. This fiber is then looped and coiled yet again.
It's not quite that pretty. It's a little more irregular than that. Pretty close. You're just getting more coils and supercoils, and it's thicker and thicker and thicker before you form a chromosome.
Leading finally to the familiar shapes known as chromosomes, which can be seen in the nucleus of dividing cells. Chromosomes are not always present. They form around the time cells divide, when the two copies of the cell's DNA need to be separated. Using computer animation based on molecular research, we are now able to see how DNA is actually copied in living cells.
Here's the DNA coming in, and it's
You are
being
looking at
split
an assembly line
and
of
copied in
major
two
miniature
directions.
biochemical machines that are pulling apart the DNA double helix and cranking out a
There's a
copy
special
of each
enzyme
strand.
there that unzips it.
The DNA to be copied enters the production line from bottom left. The whirling blue molecular machine is called helicase. It spins the DNA as fast as a jet engine as it unwinds the double helix into two strands. One strand is copied continuously
Right here.
and can be seen spooling off to the right.
The other
Things are not
side.
so simple for the other strand, because it must be copied backwards. It is drawn out repeatedly in loops and copied one section at a time. The end result is two new DNA molecules.
Remember, this only happens once when the DNA replicates itself, and that's in the resting phase.
What you are about to see is DNA's most extraordinary secret: how a simple code is turned into flesh and blood. It begins with a bundle of factors assembling at the start of a gene. A gene is simply a length of DNA instructions stretching away to the left. The assembled factors trigger the first phase of the process, reading off the information that will be needed to make the protein.
Just stopping for a second. These, the factors are called transcription factors, okay? We're going to be talking about those. Those are really important because those are the switches if sometimes it requires another gene inside the cell to make a protein, which is then a factor to turn on another gene. Okay, so there's a cascade of processes going on, and sometimes, especially embryologically, these genes don't turn on without factors that come from the outside of the cell. Okay, so that's how cells learn who they're going to be. You know, that's how we're all stem cells in the beginning. All those cells have all the genes; they can do anything they want. But it's certain factors that come in and tell certain cells that you're going to be a kidney cell, you're going to be a liver cell, and it's these transcription factors that are responsible for that. Sometimes there's a dozen of them. Okay, so and you got to have all dozen factors there before you know that you're going to be a nerve cell. Okay, or what type of nerve cell you're going to be, and that's the fascination I have with it is they're all triggers, you know. So that's the important part. And then what's called RNA polymerase is the big enzyme that comes in and basically splits open the DNA. Remember, you're not copying the DNA, you're splitting it open and making a copy of one side of it, a template. Okay, so you're putting in the opposite bases, and every 3 bases, a triplet, is what's going to code for one amino acid on this thing that will start snaking out. That's the RNA, or the messenger RNA that you're making from the DNA. That's what this process is right
now. Everything is ready to roll. 3, 2, 1, go. The blue molecule racing along the DNA is reading the gene. It's unzipping the double helix and copying one of the 2 strands.
And then puts it back together. See, there's the RNA that's being made.
The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA, letter by letter, to copy the A's, C's, T's, and G's of the gene. The only difference is that in the RNA copy, the letter T is replaced with a closely related building block known as U.
Uracil. These are called transfer RNA molecules.
Transcription in real time. It's happening right now in almost every cell in your body.
I'm sorry, no, they're not. Those are just the basic building blocks that have to go in.
When the RNA copy is complete, it snakes out into the outer part of the cell.
Through the pore. It's got to find a ribosome.
Then, in a dazzling display of choreography, all the components of a molecular machine lock together around the RNA to form a miniature factory called a ribosome. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome.
Yeah, it's called transfer RNA.
The amino acids are the small red tips attached to the transfer molecules. There are different transfer molecules for each of the 20 amino acids. Each transfer molecule carries a 3-letter code that is matched with the RNA in the machine. Now we come to the heart of the process. Inside the ribosome, the RNA is
poured where the code is being matched, but the amino acid is actually being made at the top.
3 letters at a time, and matched to 3 corresponding letters on the transfer molecules. When the right transfer molecule plugs in, the amino acid it carries is added to the growing protein chain. Again, you are watching this in real time. And after a few seconds, the assembled protein starts to emerge from the ribosome. Ribosomes can make any kind of protein. It just depends what genetic message you feed in on the RNA. In this case, the end product is hemoglobin. The cells in our bone marrow churn out 100 trillion molecules of it per second. And as a result, our muscles, brain, and all the vital organs in our body receive the oxygen they need.
Very often there'll be a chaperone sitting right there, and that protein is going into the chaperone and folding up inside. molecule, those are little oxygen. Oh, I get it. That's the mutation that causes them to crystallize like this. This is what causes sickle cell anemia. The hemoglobin sticks together and forms these long holes inside the cell, and it forces the cell from a round shape into an oblong shape. That's what sickling does. Well, that was pretty good, and that hopefully that'll give you a little bit of flavor for what we're going to be talking about here. All I did was go into YouTube and put DNA, and all those different videos will come up if you want to see the games at some point. So there's a lot of teaching power in there. Okay.
I like the visualization of DNA.
You said this is where we stopped?
Yeah.
Right there? Okay. Again, be sure you know these terms. You watch the DNA copy itself. Okay, that's called replication. The transcription is where the RNA polymerase gets on the DNA and starts to make RNA. That has to go outside the nucleus and go out to the ribosome, okay? And then that last part there where you saw all those little green transfer RNA molecules coming in, putting the amino acids together in the ribosomes, that's what's called translation. Okay? When you're actually making the protein, it's called translation. Again, here's our basic DNA structure, okay? I think you'll remember that it gets its name from the deoxyribonucleic acid. In other words, one of these 5-membered rings here in the backbone, that's the ribose there in green, right there. Okay, one of them has a missing oxygen hydroxyl group, and that's why they call it deoxy. Okay. The RNA has the hydroxyl group on there, so they just call it ribonucleic acid. That's where you get the RNA name. Okay. So they're very similar structures. But RNA is a single strand because you just made a copy of one side of the DNA, the template side. All known life uses the same bases. You can actually take human genes and put them in bacteria, and the bacteria will make the protein that the human would be making. Okay? It'll copy that. It'll make RNA from our gene. It'll send it out to the ribosomes, and they'll make human proteins. Okay? They're so close. I mean, all life is based on this structure. That's how you get insulin nowadays. They used to get it from cows, but now they do it genetically, where they have bacteria making the insulin. And there's a lot of protein-type drugs. Obviously, you can't make steroids that way, okay? But you can make proteins that way. You just take a human gene and put it in a bacteria. You can make all the human protein you want, or whatever that is. The bacteria will just make it for you because their ribosomes will read it just like ours do. Okay? So, and since we all eat, all life uses those same 20 amino acids, we don't have to worry about the bacteria missing those amino acids because they use them too. Okay, all protein is made from the same code. Okay? And so fascinating procedures. Now that the more genetic research is going on, and we now actually know the whole human genome now. That was only done in 2001. That's the first time we knew all the code for a human. And it was only one human that they did, a guy named Venter. He was the head of the research project. But now we've got thousands of humans that have had their DNA or their genome done. And now we can start comparing people and seeing what kind of mutations are causing what kind of problems and things like that. And looking for genetic diseases that we didn't know the source of before. Okay? Once you have thousands of people in the computer, it's a simple fact of letting the computer count up all the differences in the genome. Because remember, there's 3 billion base pairs in one human genome. Okay, 3 billion of those pairs. Okay? But the thing is, only about 5% of them actually code for anything. The rest of it they used to call junk DNA. It turns out that some of that junk are actually part of the switches that turn genes on, so the stuff that's right next to the gene might be useful, okay? But most of your genome is thought to be X-virus DNA. So for billions of years, we've been attacked by viruses, just like the HIV virus that puts the DNA segment in the DNA, okay? And then after, if you live, okay, eventually you get, these things get mutated and then they don't work anymore, okay? So now, but you're just building up this junk, and it's mostly viruses that have been putting it there for years, millions of years. And, but you can actually, now that we know the genome, we can go back and see what kind of virus that was, okay? And we can actually see that where back in time our species has been hit by all these viral diseases in the past. So, but the useful stuff is called a gene, okay? Just a segment of the DNA that is going to be read and make a protein, that's called a gene, okay? So I probably haven't talked about genes, but that's basically the definition, the best definition. Just a useful segment of DNA is a gene, okay? So, travel on here. Just like in that picture you saw in the first part when it was making chromatin, you saw that the DNA came in and it wrapped around that protein, okay? Those proteins are called histones. They're just little balls of protein, okay? Several hundred amino acids, but they form a little ball, a little globular protein. And they wrap around twice, each one, okay? And then they start to stock them in on top of each other like this. And the more you put together, you get a coil like this, and then you super coil that, and eventually you get a thick enough coil that you can call it a chromosome, okay? But you'll notice that in the form of the chromosome, you can't read it. You can't do anything. You have to unwind it, okay? And you have to get basically back down to this basic structure right here, and then you have to unwind it off of the histone just to read it. Because you've got to split it open to get that RNA polymerase to make the RNA. So this is an interesting little thing that's been much more recognized here in the recent past. You can change your genome, or not change the genome, you can change the expression of your genes not only by screwing around with the DNA, but by screwing around with the histones. In other words, this is something called epigenetics. I've got another slide that talks about that a little bit in the future here, but on or above genetics, okay, epigenetics. You can actually turn off a gene if it can't unwind from the histone. So chemically, you can change and react things with the histones, and you may shut down a gene, okay? Or you may take genes that are not supposed to be working and turn them on and expose them. Okay. Yeah.
I just caught like a split second of it, but this afternoon on the news they were talking about that.
Epigenetics.
Well,
or histones.
I think epigenetics. So they were talking about they were looking into being able to turn off certain genes in people to change the way they react to different things.
Sure, sure. Well, that's certainly possible, and the more we know about them, the more we're going to be doing that kind of stuff. And you may actually have genes that you want to turn on, you know, that aren't on naturally in somebody and they have a disease, say. You want to figure out how to turn it on, okay? But this is the source of a lot of what they think is toxicity of some things that are mutagenic that cause birth defects and cancer, things like that. It may not actually mutate the chromosome, or, you know, the genes, but it may affect. They think something like Agent Orange, the defoliator that was causing a lot of birth defects after the Vietnam War. They think that was what was happening with that. It was affecting the histones and not the DNA. But turning off a gene is turning off a gene, no matter how you do it. You can mutate the gene, and so it won't work, or you can just turn it off by reacting with histones. Okay.
So shouldn't you stop the cancer cells from forming that way too?
Well, if you get the right, if you do enough research, sooner or later. I don't know if I've harped on this before, but you guys, I think, are going to live long enough to see it. I may not, you know. But I'd say within 30 years, we're going to know so much about the genome, the genes, what's turned on, and what type of cancers and things like that, because every cancer is a different cancer. You know, every cancer is a different disease. It takes different genes turning on to make a lung cancer than it does turning on to make a prostate cancer or a breast cancer. And there are several types of breast cancer, and you know, different genes doing that. So we're going to find that there's probably thousands of cancer diseases, but all you have to know is which gene is being turned on or off at the right time to cause the cancer, and then you study that one disease and develop drugs that will stop it. Okay? So in the future, you're probably going to see things like, you know, you get, say, a breast cancer, you go in for a biopsy, they look at all the genes, they look at the genome of the cancer, they figure out what's wrong, and then they just go to the book and look up the list of drugs to kill that particular cancer. Okay? And it'd just be just like taking antibiotics.
Would it be different for each person?
Well, it'd be different for each cancer, but not necessarily for each person. I mean, two people could have the same mutations that cause the same type of cancer. Okay? So, but in the future, that's going to be a big deal.
What about somebody, let's just say with type 1 diabetes?
Well, they don't even know all the genes that cause that yet. See, that's part of the problem with the state of the art right now.
Is that something to do with autoimmune?
With the what?
Autoimmune?
Well, in some cases, yeah, they think even a viral infection can trigger antibodies which attack the beta islet cells in the pancreas, and then that shuts down your insulin production. So that's probably what happened to my nephew. I mean, he had a flu-like syndrome, and within weeks he was an insulin-dependent diabetic. Okay.
That's what happened to my son. He allergies. He just had a 2-week bout of allergies, and all of a sudden his sugar was 550 something.
Yeah. But not all diabetes is necessarily caused by that. Okay, because you've got hereditary, tendencies, especially the adult onset diabetes, which is much more common than the insulin-dependent, juvenile diabetes. But we don't know all the genes that trigger that yet. They just call it syndrome X, because you've got a lot of things that are happening that aren't really related. You've got things that are happening in the blood vessels, things that are happening in the lipids and the sugar regulation and all sorts of things. So there's various things that are going wrong, okay? And it would be pretty complicated to find like one drug that would cure all of that, okay? But it might be a lot easier to control it in the future, okay? So, but it's fascinating stuff, and I think if I had my whole life to do over again, I'd be going into genetic research right now. But I'll tell you a little bit of, taking animal genes and putting them in other animals, taking plant genes and putting them in animals, because we all use the same code. Even plants use the same code and the same amino acids.
They make glow-in-the-dark pigs.
Sorry?
They make glow-in-the-dark pigs.
Oh yeah.
And they made tobacco that glows in the dark too.
They've done a lot of stuff with that. They're selling glow-in-the-dark fish now. Goldfish, glow in the dark. But what they did was take the gene, okay, that makes a protein in coral that glows, it's fluorescent like that. They just took that gene and you can put it in any animal you want. You could make a glow-in-the-dark human nowadays. We have the technology. Okay. They make glow-in-the-dark tomatoes and things like that. I'll tell you about an antifreeze gene that comes from a fish that keeps them from freezing to death. It's a protein. They now take that gene and put it in tomatoes so they don't freeze in cold weather. Okay. So, because it's all the same code. All life has the same code. It doesn't matter. As long as they've got ribosomes, boom, you're making proteins. Okay, so fascinating stuff. But this is how it gets started here. So, like I said, it coils up. It's not quite as nice and neat as it was in that video. There's some, extra irregular coiling here. But eventually it coils up big enough, okay, so that you get the large structures that you call chromosomes. But most of the time, it's going to exist in a chromatin form. Otherwise, you can't read the genes. Okay? If they're all wrapped up and packaged, you can't read it. Okay? So you have to unwind it and uncoil it. Okay? So, yeah.
And the body knows when to cut it. Like in the video, I don't remember seeing like a gene or a protein that came in and actually cut it when it actually looked like a chromosome?
Yeah. Well, like the RNA, when it was making RNA, how did it know to stop? Mm-hmm. Okay. all the amino acids have 3 bases that code for amino acid. There's a 3-base code for a stop. So as soon as that RNA polymerase hits that, boom, it just, let's go. The gene's done. Okay? There's also a start, a 3-letter start code. Okay? So it knows when to start reading. Okay? Because think about it. You've got 3 bases. Okay? If you start on the wrong base, say the middle base, then you're going to be reading that one, this one, and the start of the next one. Now you're going to be, it's called a frame shift, and you're going to be totally off, making the totally wrong protein the whole way down. Okay? So those 3, you've got to start in the right place to make the right protein. But that's what genetic mutations are all about. It's knocking one of those out, or putting an extra one in, or knocking the whole thing out. Okay? And that way you get the proteins that are missing, or proteins that are wrong in the wrong place. It's bizarre, you know. And if one of your bases is A, adenine, say the other one is a T and the other one's a C, you might make a mistake in copying the RNA or even copying your DNA to get a mutation that's permanent, that would be hereditary. Say your T gets knocked out and the machine accidentally enzyme accidentally puts in another C there. So now you've got an ACC, which might be a different amino acid. Okay? And if that amino acid happens to affect, say, the site of the enzyme that's supposed to be working, if it's the wrong one, boom, that enzyme's not going to work, ever. Okay? And that's how genetic diseases work. Okay? Because, but many times, if things are going to go wrong like that, you're just never born. I mean, if you've got a genetic defect, usually you're miscarried in the first month. Okay? Almost 50% of pregnancies are. Okay? So that's it's almost a miracle that any of us are halfway normal, because everything has to go right. You know? And I think I might have told you before that all of us have about 100 mutations that didn't come from your parents. Okay? So you are really unique individuals. But what are the chances that out of 3 billion base pairs, and you could put 3 billion inches of tape all along the walls, okay, and then take 100 darts and randomly just throw them, okay? What are the chances you're going to hit one of those genes that's in 5% of that 3 billion, okay? And what is the chance that you're going to hit just the right spot that makes it not work? Okay, so you can absorb 100 mutations by random chance, pretty good. Okay, but you can also win the lottery, and so people who have genetic diseases, they basically won the lottery. Okay, because they were the 1 in a million where it didn't work out right, and that dart hit the wrong place. Okay, so but that's also how we know who's who. You can get individual DNA, and because of those different mutations, you can tell who's who. You can tell you from your parents, okay? And you can tell, because of the mutations that they had, we can tell who your parents are, okay? And we can tell who your grandparents were. We can trace it all the way back. As long as we've got the DNA, you can trace it all the way back. Because all the mutations will match. They'll be in the same places, okay? So that's how you know you're related. So if somebody wants to argue that we're not related to the caveman, the Neanderthal, all we got to do is look at the DNA and see if the mutations match, and they do. Okay, so we know we're related. Okay, here we go. A little rambling there, maybe. But basically, I think the concept, these kind of concepts, and just how this all works out, this is what you're going to remember 5 years from now, not the terms. Okay. I get to test you on the terms, but I'd be happier if you just understood how life works.
Right.
So, here's what I mean by triplets, okay? Every 3 bases in that coil, one side, they call it the sense side. This is the one that's going to be used as a template to match up to make the RNA. Every 3 bases will code for 1 amino acid, okay? And it's called a triplet when it's in the DNA. And let's see here. Don't worry. worry too much about this. This basically just shows you that you're splitting the DNA, and then you're copying each strand. This is for replication, not RNA, for replication. So you're making, so 50% of the new strands will be old DNA, and 50% is a brand new strand on each one. Okay, so they call that semi-conservative, but that's not a term you need to. This is where I would usually play that other video. I wanted to show you a picture of, this is that blue molecule, the helicase molecule, that was unwinding the DNA. Okay, it's actually got quaternary structure. It's several different proteins here that form that structure. It actually has a hole right in the middle where the DNA goes through. And this is the molecule that uncocks the hydrogen bonds. It breaks open that coil, okay? And it uses ATP to do it. It rocks back and forth, okay? And you can, there's videos out there on YouTube that show how this thing works, and how it rocks back and forth. And every time it rocks, it rips through the hydrogen bond in the DNA and opens it up. Okay? But you saw how fast it was moving. They said that was real time, and they're right. The thing is flying along there. So that ATP is flying in there. Every time it rocks, it needs an ATP. Okay, every time it moves. So there's a lot of ATP required to do that. And because it's a protein, it can have its gene mutated. Can be a problem. Okay, if your helicase doesn't work right, you get Bloom syndrome. Okay, so from now on, the rest of your life, whenever you think about anything going wrong, especially with proteins, you think genetic problems. If your ribosomes are making the wrong protein, it's because the code was wrong usually. Okay, and this is a pretty rare disease. Okay, it obviously doesn't make the helicase work bad enough that it can't copy. Okay, otherwise he wouldn't have been born, right? But it makes repair of cells very difficult. Okay, so they get cancers, they have lung problems, they have skin problems. Just even sunlight will damage the skin so much that it becomes a real problem. But it's fairly common. 1 in 100 of the Ashkenazi Jews is a carrier. That's amazingly common. Okay, for something like this. But they're a pretty inbred group. They were over in somewhere around the Caucasus, I think, around Greece and Turkey is where they're basically from. Pretty inbred group. They didn't marry. They were always persecuted and kept away from other people, and so they became pretty inbred. And when you have an inbred group like that, any mutation is going to spread through that group pretty quick. You don't have new genes coming in from the outside, diluting it out. Okay. So it's going to get more and more common. So anytime you see groups like that, like the Amish, like the Cajuns in Louisiana, fairly small inbred groups, and occasionally you'll find groups like, well, even the Finnish that have that disease, okay, Meckel-Gruber syndrome, okay. If they don't get around much, those diseases will get more and more common in those inbred groups, and so.
Dr. Irks,
yeah.
Is that why you always hear about problems with inbreeding physically?
Oh, yeah.
It has nothing to do with the fact that because that's what it has to do with. It's the fact that you're not getting the.
Yeah, that's the fear.
Okay.
Sisters marrying brothers and things like that. It doesn't happen as often. The child usually doesn't have a problem.
Okay.
Okay. So that doesn't happen quite as often as people think it does.
Okay.
And with cousins and things like that, there's always a chance of things like this happen. But it's only like 1 or 2% of consanguine siblings that have children with problems.
Okay.
So the chances are fairly small.
But is that essentially why, though, is for that reason?
It's the genetic stuff you're worried about.
Okay.
Because if your parents are carriers or something, then you've got a 50-50 chance of having it. Your sister has a 50% chance of having it. So you got a pretty good chance of giving something really nasty to a child if that's the case.
Gotcha.
Okay. So, and it's less of a chance with cousins, but it's still a higher chance than the other population.
Okay.
But in a lot of societies, cousin marriages are real common. Yeah. Well, before I get off the subject, there's a lot of fascinating neurological diseases that the Amish have, because there's about 150,000 Amish, I think, at least as a couple of years ago when I was reading this article, but they all pretty much descended from about 100 couples. So they're a pretty inbred group, and they have a lot of rare neurological autosomal recessive disorders, things you won't see in the rest of the population. But on the other hand, those original 100 people may have had less of an instance of something else that's bad. Okay, so the inbreeding effect can sometimes protect you from things as well as hurt you. Okay, maybe they don't have as much hardening of the arteries as normal people because the people that survived that trip were healthy and blah blah blah. You know, sometimes that founder effect, they call it, is good for you, but other times it's not.
So by inbreeding, you don't necessarily mean sisters and brothers and all that.
No,
you mean a
within a group
with nobody else. Like 100 people could not be related in any way.
Right.
But because they don't bring anybody else in,
Right.
eventually they'll all be related.
Right, that's exactly right.
Like all of
Yeah.
us.
You're keeping the gene pool.
Mm-hmm.
Yeah. I mean,
Who wants to?
well, as humans, okay, you can look at it as humans, we're inbred. Okay, we all have the same kind of diseases because we're keep giving them to each other, blah 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 blah. You know?
Right.
And we are more inbred than, say, chimpanzees. They have much greater diversity in their DNA than we do.
They don't have the same social pressures that we have.
No.
You know.
Stop replicating.
Exactly.
Well, what happened was, say you have a large population of chimps, or a population of chimps and a population of humans, okay, that live in the same area. Over time, over a long period of time, you're going to get a fair amount of diversity in those groups, okay? But what happens is, one part of this group wanders off to the Middle East and over to China, and they're not nearly as diverse as the group that stayed behind, because they don't have the diversity of the whole group. That's why there's much less genetic diversity in Caucasians and Asians than there is in Africans, because it's a much older group. You can trace the mutations all the way back, and the younger groups that came out have much less diversity. Okay? So, and they've actually traced it. They found the village in Africa, East Africa, right by where they found Lucy, you know, the ancient hominid. That village has more diversity even within the village than all of America does. There's more differences, and so it's a healthier group, basically. Okay? More diversity is better for you, because it protects you from all these inbred problems. So. Well, think about it. Just with skin color. I mean, the group that came out had a mutation, okay, that knocked out the melanin-producing melanocytes, and then all of a sudden, that group is now all inbred. I mean, so that's how it spread. So,
Is it locked?
It should be locked up in the unit.
okay. Let's move on. DNA polymerase, just burn that into your brain. That's the one that copies the DNA, okay? And that's about as complicated as a question as I will ask, okay? What is DNA polymerase? What does it do? Copies DNA. Here's a little PBS moment. You can screw around with the DNA not only by picking and putting in extra bases and things like that, and knocking out genes and making genes backwards and things like that, but here's what keeps me awake at night. This is the major carcinogen in tobacco smoke. See these aromatic rings?
Mm-hmm.
Well, it goes through the peroxisome. It gets oxidized in this position. That's a very highly strained little three-membered ring, which will pop open real easy to form covalent bonds. Okay, this thing gets jammed into the DNA and forming covalent bonds with the bases, and it's basically, you can see that helicase come whipping through here, and that's like putting a stick in the spokes. Okay, so you can see how it's going to make mistakes when it hits these things. Okay, you're going to get mutations. Okay, that's what causes the cancers.
I have a question for
Yeah.
you.
Yeah.
I've wondered this since we started the class. With all you know about this,
Uh-huh.
how do you continue smoking? I've just been curious.
Yeah, well,
I
you're
mean-
right, you're right. Who can explain addictions though? Yeah. I mean, I've tried quitting a couple of times,
Yeah.
and I'm miserable, and I
Well, I
make people
quit
around me
16
miserable.
years ago.
Listen,
Just
my
because of the
grandmother-
aging of the skin.
My
I mean,
grandmother
Yeah.
had
you know,
lung cancer.
my,
and she still
my,
smoked.
my grandmother is 93, and she looks younger than my father because she doesn't smoke, drink, and my dad did all of it. Still drinks, but we took my dad out for his birthday, and nobody believed that she was his mother. They thought maybe sister, you know. And,
Yeah.
but she quit
Makes a difference.
smoking years and years. She maybe smoked a couple years and just didn't like to spend the money on it.
I've sort of given up. I just tell people I don't want to live forever, so.
It's really a lot harder than it looks though, because I was gonna say, my grandma had to get she had to have like a lobectomy done. She had to have a little bit of a right lung removed, and she still smokes. She quit for 3 weeks, and she still smokes. It's just as bad as before.
Yeah.
But now that leads me to another question.
Stronger wills, we gotta want it.
The fact that this is always happening, is that why when someone who's quit smoking, they say that their lungs can heal themselves? Is that why? Because this is always...
No, they don't heal themselves
They
completely.
don't, okay.
No, there's damage to the lining and things like that. There's scar tissue, basically.
Well, how much truth is there to the healing itself part?
Well, occasionally when you smoked 20 years, most of that respiratory epithelium is gone.
Really?
It is now stratified squamous.
So you've already...
Like skin on the...
So if I smoked for
Well,
15
I tell
years...
them, it's their choice. Just tell them,
Yeah.
what the problems are caused. Just like anything, drinking or drugs or anything else, or
Right.
driving a car
Or the
fast,
damage
just
you
take the
do.
risk.
So if I smoke for 15 years but haven't smoked for over 10, it's not going to reverse itself. There's still going to be permanent damage there from the years I
Yeah, you
did.
can still tell they're a smoker when they do an autopsy when they're 80.
Really?
Yeah, yeah.
Wow.
But things like this don't necessarily go away, but it takes years and years and years of getting hit by these things to get hit in the right place.
Okay.
See,
So you
most
can
of that
never know that.
95% of the gene or the genome doesn't care. You can skip over that and make a mistake, but it's not going to cause a cancer.
Okay.
What you have to do is hit the transcription factors usually,
Okay.
to turn things on or turn things off that shouldn't be on or off.
Okay.
So
And that's what causes the cancer?
yeah. Usually it takes several mutations, usually about 3 or 4 mutations in the right genes to cause a cancer. Okay. So you can, it takes years and years and years for those hits to, it's a probability factor. Okay? Some people are lucky. I mean, my father-in-law smoked for 60 years and died at 92, so
Yeah.
from a broken hip.
Mine's gonna be really silly probably, but okay, you're saying that you can break off DNA and cause mutations and stuff and then reading genes.
Yeah.
Okay, can you do Spider-Man kind of stuff with putting
Yeah.
make somebody run faster by inserting a certain
Yeah.
thing or
Yeah, you could. If you want to do genetic engineering like that, yeah. You can give them all fast twitch fibers and make a sprinter out of it.
make us be
You
able
know
to
the
breath
genes to do
longer,
that. Okay?
underwater.
The sprinters are a lot different than the long distance runners. They have different types of muscle fibers. Okay? They have different numbers of mitochondria, different glycogen stores, and all sorts of things. The muscle fibers are different. Okay? But one fascinating thing that they're doing now is they take the gene that codes for the silk from spiders, and they did goats. They put it in goats, and now the female goats, their milk is, they get the fiber out of the milk. They get the silk out of the milk. Did you hear about this? So they can make silk now from goats.
Oh, that's cool.
And so this is the kind of stuff that's just going to be
So
swamp in the
saying
future.
Now that's very cool, but at the same time, it's kind of creepy. Yeah.
Well, so if they put that gene into the goat, they do it in the fetal goat, or
Well, they have to do it in the embryo.
the
Yeah.
embryo? That's what I meant. Before it's ever born,
Right.
they insert it, and then
Yeah.
that goat, when it's born, that happens.
Yeah. They have to put it in the egg,
Right.
and then it grows up.
Right. So the one that they put it in, so it doesn't have to go like a couple generations before
Yeah,
it starts
yeah,
working.
yeah, yeah.
You just put it in the
Well,
embryo.
and I don't know enough about it to really be, talk
Right.
expertly, but what I wondered when I heard that was, how did they know it was gonna be in the milk?
Right, that was
How
my
did they
other.
not know that it was gonna be in the saliva or
Maybe
something?
they did.
Right.
Well, they might not have,
Yeah.
but they obviously got lucky and now it's producing in the milk, but, do you know anything about that? How they guide that?
No, I'm not the same as you on that one.
Yeah, okay. Because, you can think about a lot of ways that things can go wrong. Okay, so, or why not say the kidney cells were producing the silk and killing the kidneys and they were dying?
Right.
I don't know, but,
Maybe they did. I mean, if you can do mutations, why can't you do mutations to make it better, instead of
well, you
all
can.
these mutations?
You can, but you'd have
not
to do it
legal,
in
is
an
it?
egg.
That's the problem.
Well, that's where stem cell research is coming now, though,
Yeah.
because they're trying to do it with stem cells. But
Right, you're trying to correct
it's easier to
things.
do it when you're first starting as a single egg to reproduce.
Right.
And this
Right, because
now-
remember, when you have a genetic disease, every cell in your body has the same
Right.
problem. Okay? So if you're gonna fix it, the time to fix it is in the egg. When you've only got one set of chromosomes. Okay? There is a report now where they have done some gene therapy with some of the diseases that are genetic, or at least show, chromosomal damage to cause something. There was a childhood leukemia that I think they declared that they cured this little fellow of it. They took the gene that he didn't have that wasn't working, they put it in a virus, like a cold virus, and they gave it to him. Okay, the virus obviously attacks the cells and inserts the DNA into the cells, and it cured his problem. Okay.
Wow.
So, but that's like the only case I know of, but that's what they're trying to do now. Okay, if you can find the genetic problem, you have to have a carrier to get the gene in if you're talking about treating adults. Okay, so you've got to put the gene in there somehow. Okay. Okay.
So as fast as our bodies produce, different, yes, cells and they replicate.
Yeah.
So if you insert something like that within a matter of what, a day, 2 days, you would see improvement?
Oh yeah, the virus will get in just like a cold virus. It wipes out your respiratory lining in hours. Okay, and then you're growing new stuff. But the object is not to kill the cells with this virus, and it doesn't kill the cells. It just inserts the DNA and then says, I'm done.
Just
Okay?
mutates them, basically.
Yeah.
Or changes them.
So all those cells, when they divide now, will have correct genes. Okay, because they'll copy the good one.
Right.
So from then on,
Is that why they recommend, like, when they freeze the placenta, the cord blood?
yeah,
Is that
exactly.
some of the reason
Yeah,
why
the
they
stem
do
cells.
that?
Yeah, because you can tell those cells to do anything.
Right.
They could be nerves, skin, heart, lung, kidney, whatever. They'll do anything. That's what stem cells are for. But there's still a ways to go. We got to know how to control these things, how to work the transcription factors, and but it's doable. Okay, but it's going to be 20 years, 30 years. I think you guys are going to, when you're my age, you're going to be living in a different world when it comes to medicine. Okay, so.
You said that they had the kid that they gave the virus and it fixed something. Is that how, you catch the virus in that Guillain-Barré syndrome or whatever, how it attacks all your cells? Or?
Well, flu viruses will attack your respiratory tract. They don't, like a flu virus is not going to attack necessarily your kidneys or anything like that. It attacks certain types of cells because it has receptors. So it's guiding it to where to go. It knows where to go in the respiratory tract. So it's getting that pseudostratified columnar epithelium. That's what it attaches to. But what it triggers, what any kind of virus can trigger actually, is an antibody response, and the antibodies is what attacks the nerves and causes Guillain-Barré syndrome.
So your body is
paralysis.
itself.
Yeah, it's just like getting rheumatoid arthritis against your joints. Okay? It's antibodies attacking your joints.
The patient we had said it happened within an hour. He went from fine until his whole body was...
Yeah. I've had 4 or 5 friends that had it, so it's a pretty common disorder. Several doctors had it, had to quit surgery and all sorts of things because they had paralysis that persisted. One guy was on a ventilator for a month. But and I saw several cases in training. You don't tend to see it out in the clinic, but you're going to see it in the ER. And you're right. People wake up one morning after a cold. Usually that may be a week ago or 2 weeks ago. But they wake up thinking, "Wow, I'm really weak. I can hardly move my legs, my feet." And this is what happened to my friend. He got on the phone by the time the ambulance got to him and got him to the hospital. I'm sorry?
Did it just happen recently?
Oh, no, this was a couple years ago.
Because this...
Not here. He was up in Idaho.
The one I'm talking about is a doctor.
Ah, who?
I can't tell you. You're trying to get me to break the HIPAA. You're trying to make me lose my job.
It's been a long time. It's been a long time.
Well, this happened like
If I knew, I'd tell you. Well, I'll have to check.
It happened last year. So, like he said, like he said, by the time he called the ambulance and they got there, like he
was paralyzed.
Paralyzed.
Yes, was on a respirator on the way to the hospital.
Yes.
So it can happen quick. Well, we need to get through some slides here.
Yeah, let's do it.
We need to get through some slides here. Okay. We talked about genes. Okay. Not all of the gene is actually useful. Okay. You can have good parts and bad parts. The good parts are called exons, and introns are the parts that you don't need. Okay. And so the gene is going to be copied, and you're going to get the RNA for the whole strand. But some of that RNA has to be spliced out. Okay, and that's an interesting process. The enzyme that does it is called a spliceosome. And look at it this way. I think I can visualize it because I don't have a good slide or a model of it. But here's your RNA that's been made from a gene. The part that's my hands, let's say, is a part I don't need. I want to hook my wrist to my wrist, and that's the gene. That's the RNA that I want to make a protein with. So what it does, it loops up the part it doesn't want, and then snips it off and connects the wrist, and then this part just leaves. So that's why they call it a spliceosome. Okay, that will do that. This is how you can make random antibodies that are proteins, and you can make millions of different kinds. We only have 23,000 genes. So if our immune system relied on 1,000 genes, we would all be dead. Okay, we need millions of different kinds of antibodies that fight off different kinds of organisms, and this is how your immune system can do it. It takes a gene from column A and a column B and a column C, and it puts them together and then splices out different areas. Okay, so everyone is randomly different. That's why some people would be immune to some disorders and others wouldn't, because you don't have the random antibodies that are going to protect you where somebody else might. Okay, and usually in a population, you're not going to kill off more than maybe 90–95% of the people. You're actually going to find people who are immune to whatever Ebola or whatever disease you're going to give them. Okay, even AIDS doesn't kill everybody. Okay, sorry. Well, he's taking drugs, but they think about 10% of the population of Europe is immune to AIDS. And I think that's because of something that happened. It was a very healthy sort of select group that survived the Black Plague, and they think there are some antibodies that are in common, okay, that fight off the AIDS virus.
So then they need to take their DNA and give it to all of us.
Well, that would be one issue, yeah, or at least find out what antibodies are helpful, and they're studying that right now.
And you said spliceosome?
Spliceosome. I don't think I've got it written down, but I don't even remember if I put it on the test in the past, but that's the idea. Okay. These are the kind of things that we were just talking about. Point mutations, deletions, insertions, duplications, okay? So I don't want to spend, you put a gene in the wrong place, okay? This is during meiosis, when you're getting crossover of chromosomes between mother and father, okay? That's why you inherit only one chromosome from your mother, but it's a 50-50 mix of her parents, okay? That's why you're not just getting one chromosome from your grandfather passed to your grandmother to you, okay? During meiosis, the chromosomes will actually flip over each other and split and switch. They'll switch, say 50% of the chromosome goes to one and the other goes, and then they split up again, okay? Then they do that again, okay? That's called crossing over. That's how you get a blending of your mother and father even in the one chromosome you're going to give to your child. Okay? Otherwise, you'd be giving them either your father's chromosome or your mother's chromosome, and there'd be no change. Okay? So that'd be like your grandfather and your grandmother on the other side having a kid. Okay? It'd be the same chromosome moving down, right? You have to cross them over to get that 50-50 mix. Okay? That's what happens in the egg, in the meiosis. Okay? And the sperm.
And that's the translocation or the inversion?
That's the, well, that can cause translocation problems. In other words, when it crosses over, somehow the gene gets in the wrong place, and say now the transcription factors, the switches aren't there.
Okay.
Okay, so you could cause a problem that way.
Okay.
One interesting thing, though, is the duplication. You can switch over and say those two genes on the same chromosome, they should be relatively matched. Okay, your mother's gene for hair color and your father's gene for hair color, they switch over at that site, and what if they split in the wrong place now, and now you've got two genes for this hair color, and this chromosome has none. Okay. That's what I mean by duplication. You've duplicated that gene twice now on the other chromosome. So this is how you get tricolor vision. Okay? We have visual proteins that are sensitive to light, mostly black and white. You duplicate the gene, and then over millions of years you start mutating it, and it absorbs light at a different frequency. Then you start to see colors, different frequency. It does that 4 times, and now we see in 3 colors. Okay? That's how you get it. Most animals don't have that. Okay? Like, that's why your dog is colorblind, essentially, because he doesn't have all that tricolor vision. Okay? But most of the primates do, the higher primates, because we all have the same mutation in the same place, so we know we're related. But other animals, like pigeons and shrimp, have much better vision, much better color vision. They have like 7 of these things. So they're seeing in a really broad spectrum. This is how bees see in the ultraviolet, okay? Because they have the duplication, and now that it was mutated, and now they see in the ultraviolet light range. Just like whales and porpoises, they see in the ultraviolet, okay? Because they have the mutation, and it went far to the blue range. So we see blue, they go far into the ultraviolet, okay? So they can see underwater better.
I have a quick question.
Yeah.
If, like, in eyes, how can you have like 2 colored eyes if brown is a dominant gene, and then how can you have like a brown eye and a blue eye? Like, what happens? Is it a mutation?
I'm sure, some kind of heritable thing, but yeah, that's a good question. I don't know the exact answer, but what is it? Anisocoria is when you have 2 eyes, different colors. But I forget the exact mechanism. Okay. It might just be that, yeah, one eye, the gene is turned off. The dominant gene is turned off by mistake. Because blue is sort of a fallback eye color, because blue is not really an eye color. Your eyes have fibers in there that reflect light. They absorb the yellows and the greens and reflect the blue, just like the sky. That's why the sky is blue. Okay? So your eyes are not really blue. They're reflecting blue light. Okay? It's not like your shirt's blue. Okay? It really is blue, but your eyes aren't blue. That's why blue eyes don't look so blue when you get in gray light, because they're not reflecting the blue. Okay? They look gray.
So what color are your eyes then?
Clear.
Well, the brown is a real color. Brown is melanin. Okay?
Right.
What about hazel?
And the less melanin you have, the lighter color you're going to be. And if you have certain types of melanin, and you can mix and match and get green eyes, but the blue, the green eyes comes from the blue and a slightly yellowish pigment in the eye, so you get green eyes.
So green really isn't a color either.
Right.
Okay.
The blue part isn't.
What
so
about
really it's just brown.
hazel eyes?
In darker light, their eyes are going to look more yellowish.
Right.
Okay.
So what about hazel eyes? Is that why they change color? Is the
Yeah. So
Well, what about if your parents have
I know it.
brown eyes and you have blue eyes?
that's just luck of the draw.
That's the milk brand.
Luck of the draw. What you got?
But like if your grandparent, is it because maybe your grandparents have?
Somebody, to have blue eyes, you have to have the right size fibers. Okay, so that's hereditary. But if you don't inherit enough of the melanin-producing genes, you'll have blue eyes. So it's just a matter of chance from the crossover. So I'm not going to. You don't have to memorize this list. okay? But I might say something real simple, like genetic mutations consist of things like deletions and insertions. True or false?
True.
Okay, something real simple. Okay. Ice fish. We got 15 minutes. Since we saw that video, that really helped speed things up as far as I think your concept of what's going on. But I don't want to spend a minute or two on this one, right? But this was a PBS moment. This is my favorite animal in the whole wide world. This is the most unique thing I've ever seen. It's the ice fish in Antarctica. They live in sub-freezing cold waters, -3 degrees, where other fish die because their blood freezes. Okay. These guys, but the benefit of living in that water is that there's no competition for the krill and other things that they can eat. But there's more oxygen in cold water. Okay. So what happened was, these guys had a mutation where they lost their scales. They are able to absorb oxygen through their skin, so they really don't even need gills, although they still have them. Okay, so they get they benefit from that higher level of oxygen in the cold water. It comes through their skin. Then, apparently, if only that happened, they'd still have to kind of live on the edge of the cold water, right? Because they'd still freeze if they went into it. They have a mutation in a protein. It was like a gene that was read backwards or something, and it turned out to be fantastic because it stops ice crystals from forming in their blood. Okay, it's like a little protein. As soon as the ice crystal starts, just the hydrogen bonding, the protein will wrap around it and stop it. Okay, so they can now live in the sub-freezing water like other animals can't. Okay, and then the latest thing that happened was they had a mutation that knocked out the hemoglobin in their blood, so they couldn't carry oxygen with hemoglobin. But they didn't need it because the oxygen was just coming through their skin. So it didn't kill them. Okay, so they have no blood.
Oh, wow. Wow.
It's the weirdest animal I've ever heard. Okay?
And that's why it's your favorite.
Yeah.
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 This is weird.
That's, okay. Well, epigenetics is all the stuff we were just talking about. You're screwing around with those histones. Okay? So you've got the slide. I don't need to talk about it too much anymore. Okay. I'm trying to finish up here so we can do like a big review next time. This is just a slide, not as good as that video, but here's your transcription factors. Here's the switches. Okay. When they all line up in front of the gene, here's the start of the gene, the DNA. Okay. The RNA polymerase is the name of the enzyme that's going to sit right there and then read that DNA making RNA. Okay. Now remember, it's the DNA polymerase that copies DNA. It's the RNA polymerase that makes RNA. Okay, and that's about understanding that and understanding what transcription factors are, how they're switches. Okay.